Okay, now we're recording. Sorry about that. Let's go back to the PowerPoint. Okay, so again, KLH stands for keyhole. Limpet. Cyanogen. You don't have to know this. If you just, on the test, if you just know KLH, you'll be fine. And I know it's really hard to read my writing when I'm trying to write with a mouse. Keyhole limpet cyanogen. So a keyhole limpet is a little snail-like marine creature, and it produces this one protein that's extremely immunogenic. Okay, so if our protein is small, not very immunogenic, we can covalently conjugate it to a carrier protein like KLH, and KLH will make it more immunogenic so that we can get an immune response against it. Okay, so we can do this with small proteins. We can even do this with small peptides, even small molecules called haptens. So if you have like a small organic molecule, I'm talking maybe something with a benzene ring and not much more, by conjugating it to a carrier protein like KLH, we can actually get an immune response against something as small as a small organic molecule. Okay, so getting back to how we make these antibodies. So we take our protein of interest. We might have to conjugate it to a carrier protein. Definitely have to add an adjuvant to enhance the immune response. Okay, then we have a regular immunization schedule. So we'll start immunizing rabbits on a regular immunization schedule, and then we'll schedule bleeds every so often. So after we bleed the rabbit, we'll assay the blood. We'll look for antibodies specifically against the antigens, well, I should say epitopes contained on our protein. And then I think at about day 28 of the immunization schedule is the terminal bleed. We'll just take all of the blood from the rabbit if we're getting a good immune response. So then what we have is something called a polyclonal antiserum. Okay, and there's a difference between a polyclonal antiserum and a monoclonal antiserum. Let's talk about that. Okay, so continuing, so blood is drawn at regular intervals and centrifuged to remove blood cells from the serum. The remaining supernatant is called the antiserum. And the antiserum is what we're going to use to detect the protein. Okay, now we can have two kinds of antiserum. We can have a polyclonal antiserum, or we can have a monoclonal antiserum. So if we do a protocol like I showed you with the rabbit, what we're going to get is a polyclonal antiserum. So when we inject a rabbit with an antigen, like a purified protein, many of the rabbit's B cells are going to be primed by that protein to start producing antibodies. Okay, so once these B cells are primed, they're activated, they're going to differentiate into, plasma cells that can make a lot of antibody, and also they're going to divide. So each B cell that becomes activated by the protein is going to proliferate into a line of B cells that make one particular antibody that recognizes one particular epitope on the protein. Okay, so let's look at the figure here. So remember the parts of the antibody, so the ends of the Y here, these are called paratopes. They're the sticky part of the antibody. And the part of the protein that's recognized by a particular paratope is called an epitope. So each of these antibodies comes from a different B cell line. each B cell line is going to produce one antibody that recognizes one particular epitope. Okay, so if we have a polyclonal antiserum, we have lots of different antibodies from different cell lines that recognize lots of different epitopes contained on the protein that we immunize them with. Okay, so a polyclonal serum, I'm reading from the second bullet point, a polyclonal antiserum will contain antibodies produced by many different lines of immune cells, specifically B cells. Thus, a polyclonal antiserum will contain many different antibodies directed at different epitopes. So situations where we would want to have a polyclonal antiserum rather than a monoclonal antiserum is, say we're trying to detect a protein that's present at very low levels. Okay. So the more antibodies we have that recognize different epitopes, that's going to boost our signal. So more antibodies binding to that protein is going to make it easier to detect something that's present at low levels. Okay, if we had a monoclonal antiserum like this, and this protein were present at low levels, you can imagine that it's going to be harder to detect because we don't have as many antibodies, I should say, binding to it. Okay. But there are some situations where this having a monoclonal antibody is desirable. And we can make monoclonal antibody lines. Okay, now making a monoclonal antibody is, it's a little more complicated, but it's doable. So usually, rather than using a rabbit for polyclonal antisera, we would, like we do for polyclonal antisera, we use a mouse for making monoclonal antisera. Okay, so this part is just like what we do with the rabbit. We take our protein of interest, we're going to mix it with an adjuvant that enhances the immune response. Again, if our protein is hypoallergenic, we're going to want to conjugate it to a carrier protein to boost the immunogenicity. And then we inject it into our mice on a regular immunization schedule. After a certain amount of time, the mice are sacrificed, and the spleen is dissected out, and then the spleen is, so we break down this, we extract B cells from the spleen of the animal. So the spleen is an organ of the immune system. So it will contain B cells that have been primed, basically. They've been activated by exposure to an antigen, specifically the protein that we want to raise antibodies against. All right, so spleen cells, these are an example of primary cell culture. And when we take cells directly out of an organism or a tissue, that's what we call a primary cell line. If we tried to grow these cells, they have a limited number of divisions before these cells are going to stop dividing and they're going to die. Okay? How can we fix that? We don't want these cells to stop dividing, because if they're making the antibody we want to be able to make that antibody forever, right? So how do we do that? So we take myeloma cells. Myeloma cells are an immortalized cell line. Okay, these are cancerous cells. Cancerous cells don't stop dividing after a certain amount of divisions. They can divide and divide and divide indefinitely. So if we take myeloma cells, mix them together with our activated spleen cells from our mouse, and we're going to make what's called hybridomas. We're going to fuse them together into hybridomas. There's a couple ways this can be done. We can do this chemically so in this example, we're using polyethylene glycol. This is a chemical you might have heard of before. It's usually abbreviated PEG. Okay, so chemically, by adding PEG, we can cause the hybridoma cell, we can cause the myeloma cells, rather, to fuse with our mouse cells and then make what's called a hybridoma cell. Hybridoma cells are immortalized. and they can also make antibody. They're a fusion of the two cells. So these cells can divide indefinitely, and they can still produce our antibody, which is what we want. So in this example, they use PEG. You can also use a technique called electrofusion. So you can use electricity to make these hybridomas. Or you could use a virus. There are certain viruses that will fuse the cell membranes of cells. But regardless of which technique you use, once you make your hybridomas, they will divide indefinitely in culture and also make antibodies for you. So when I was in graduate school, we had a monoclonal annex where we made monoclonal antibodies. So typically, rather than growing them in a flask like this, what you would do is dilute out your hybridoma cells. Oh, sorry, I'm looking for this. So you would dilute your hybridoma cells and then grow them in 96 well plates. So 96 well plates, they kind of look like an ELISA plate. Let me show you an ELISA plate. Okay. This is a 96 well ELISA plate, but they make 96 well cell culture plates that look a lot like this. So what you would do, what we did when I was in graduate school, is you dilute out your hybridoma cells, and so that and then you put them in the 96 well plates. They should be dilute enough that on average, you're going to get one cell per well. Okay? Then, of course, these hybridomas are going to grow, they're going to multiply, and they're going to start making antibody and secreting antibody into the growth medium. So every so often, you can take a sample of the growth medium, and then you can test that to see if it contains antibody against the protein that you raised the antibody against the protein that you injected the mouse with. Okay, and you can also test it to see exactly. Remember, we're just gonna have one epitope recognized by these monoclonal antibodies. So you can also test to see exactly where on the protein the epitope is located that's recognized by that monoclonal antibody. Okay. So going back to the bullet points quickly. So first bullet point, mice are injected with a protein of interest and an adjuvant, and an immune response is elicited. Antibody-producing cells are isolated from the mouse's spleen and fused with multiple myeloma cells. Okay, so this immortalizes them so they can divide indefinitely. These are called hybridomas. The hybridomas are diluted so that we only get like one hybridoma per cell, and each hybridoma is then going to produce a monoclonal antibody. Specific hybridoma lines are used to grow monoclonal antibodies. Okay, so bottom panel here. Top panel, this is what happens with a polyclonal. Down here, this is an example of using a monoclonal antibody. So we have one antibody derived from one cell line, and it just recognizes one single epitope on our protein of interest that we immunized our mouse with. So monoclonal antibodies are produced by a single line of immune cells. Thus monoclonal antibodies are directed at a single epitope contained on the antigen, so down here. The specificity of monoclonal antibodies makes them useful in clinical assays, like for heart attack, HIV. Also for pregnancy tests. Home pregnancy tests use monoclonal antibodies that are directed against a hormone that's released once the egg implants in the uterus. Okay, it's called, this is a hormone called human chorionic gonadotropin. Antibodies against human chorionic gonadotropin, or HCG, are used for home pregnancy tests, and these are actually very accurate, using monoclonal antibodies. Cancer therapies. So monoclonal antibodies can be raised against cell surface proteins that are only expressed on cancerous cells. In some case, the antibody is enough to direct your immune system to get rid of the cancerous cells. Sometimes we can attach either drugs or radioactive isotopes to monoclonal antibodies and use them as a drug therapy. So this way, the monoclonal antibody will be directed towards cells that express this surface determinant that only cancer cells express. So the monoclonals will be directed against the cancer cells and specifically either target the drug or target radiation, in the case of isotopes, to tumor cells. Okay, so the monoclonals will ignore healthy cells, target the tumor cells, and carry drugs directly to those tumor cells. Purification of scarce proteins. So when we talked about purification of proteins, one very powerful technique for purifying proteins is affinity purification. So we could use a monoclonal antibody. We could use the binding properties specifically of a monoclonal antibody to do affinity purification. So we could take a monoclonal antibody, we can covalently attach it to beads in a chromatography column, and then we could use that to do affinity chromatography to purify proteins. Okay, so let's talk about, so I talked a lot about making these antibodies. Let's talk about some of the techniques where we can actually use antibodies to detect proteins. Pardon me a second. Okay, one technique we can use to detect proteins using antibodies is called ELISA. Okay, ELISA is an acronym for Enzyme-Linked Immunoadsorbent Assay. Okay, so Enzyme-Linked Immunoadsorbent Assay, or ELISA. There's a couple different kinds of ELISAs. ELISAs are very useful. We have, hopefully we'll get to use our ELISA reader that we have in the research lab. We have a nice ELISA reader. I do plan on, I'm trying to think of what I ordered. I would like to do quantitative ELISA with you guys. I don't know if I ordered the right materials for that. But we will do it ELISAs later on in the semester in lab. So there's a couple different kinds we can do. We can do an indirect ELISA. We can do sandwich ELISAs. First, let's talk about indirect ELISAs. So an indirect ELISA can be used to test for HIV infection by, sorry, there's a lot of typos in this, by detecting antibodies directed against HIV antigen. Okay, so in an indirect ELISA, we take the ELISA plate, and the first thing we do is we coat the ELISA wells with antigen. Okay, so again, before I go any farther, so remember that an ELISA plate looks like this. Okay, they're 96 well plates. They kind of look like a 96 well culture plate, like we would use to make, to grow up the hybridoma cells. But this is an ELISA plate here. Each one of these circles is a well. And here in this diagram, each one of these represents one of those 96 wells that I showed you in the ELISA plate. Okay, so in an indirect ELISA, the first step is to coat the wells with antigen. So we take the protein that we want to detect. Well, that's not entirely true. What we're assaying for in an indirect ELISA, we're not really looking for the protein. We're looking for antibodies that recognize that protein. So in an indirect ELISA, the first thing we do is we coat the wells with the protein that's detected by the antibodies. Second step, so we do that, then we do a wash step to wash away any impurities. Second step is when we add our human serum. So let's say, for example, that we're trying to detect antibodies directed against HIV. Okay, so we can tell with an ELISA like this, we can tell if somebody has been exposed to HIV and their body has produced antibodies against it. Okay, similarly, let's say, well, because we're in a pandemic, we could do this with COVID-19 too. We could, instead of using HIV proteins, we could purify the spike proteins from the coronavirus that causes COVID-19, and we could coat an ELISA well with that. Okay. Next step, we take patient antiserum. Okay. So we take, so we draw blood from a patient who's either been exposed to HIV, like in the first example, or maybe has possibly been exposed to the coronavirus in the second example. Draw their blood. We spin down the blood cells in a centrifuge. The liquid that's left over is serum. So we take that serum, dilute it, and add it to the wells. Okay? If it contains antibodies that either recognize like HIV, like in my first example, or say they recognize coronavirus proteins, like in my second example, they're going to stick to them. Okay? However, we can't visualize this yet. In order to visualize this, we need a secondary antibody. This is where the indirect part comes in. Okay? So again, in both my examples, we're treating human patients. So we take human blood and we incubate the human serum with the antibody, with the antigen that the antibody detects. Okay? We need a secondary antibody. So typically, when we're doing ELISAs, the second antibody comes from a goat. So what we can do is we can purify human antibodies and then inject them into another species. Usually we use a goat for a secondary antibody. So when we inject human antibodies into a goat, the goat's B cells recognize human antibodies as something foreign. So the goat starts to produce antibodies against the human antibodies, right? So now we have an anti-antibody antibody, if that makes sense. We have a goat antibody that will specifically bind to human antibodies, okay? So we take that goat secondary antibody and we conjugate it to an enzyme that we can detect, okay? Commonly in these experiments, the enzyme that we use is horseradish peroxidase, or HRP. Okay, HRP, let me write this down. That's horseradish, just like the condiment. Peroxidase. Oh, it's awful messy. That's supposed to be a P. It's supposed to be an E. Okay, that's horseradish peroxidase. Okay, peroxidase, just like the peroxide, except spelled a little differently. Horseradish peroxidase, or HRP. Okay. So in the second step, we incubate our secondary antibody with our primary antibody. If there is antibodies from the first incubation that are adhered to the antigen proteins, then the secondary antibody is going to bind to them. Okay. And the secondary antibody is bound to an enzyme. Okay. In the last step, we add a substrate that the enzyme can react with to produce a colorimetric reaction. Okay, so the third step, we add an enzyme. We add a substrate, If the enzyme is present, it's going to convert the substrate and cause a colorimetric reaction. And in this way, we can detect whether or not there are antibodies present in the human serum that recognize the antigen. This makes sense to everybody? Okay. So actually, it's really simple. So indirect ELISA, in indirect ELISA, one thing to remember about indirect is we're not looking for the protein, we're looking for antibodies against that protein. Okay, so the first step, we coat the wells with the protein. Second step, we incubate with human serum that may or may not contain antibodies against that protein. Okay, this first step is called so this is called the primary antibody. That's the antiserum that may or may not contain the antibody. Okay, then we incubate with secondary antibody. So the secondary antibody, in this case, is goat anti-human antibody that's conjugated to HRP. Third step, we add a substrate, and the enzyme will convert this substrate into a product that will give us a colorimetric reaction. Is everybody good with indirect ELISA so far? Okay. Okay, so here's an example of what an ELISA looks like. So in this ELISA, one thing you can do with ELISAs is these can be quantitative, meaning that you can not only can you detect whether or not a protein is present or whether or not an antiserum contains an antibody directed against a certain protein. You can quantify them as well. You can figure out how much of that protein is there or how much of that antibody is present. Okay, so I think in this is an ELISA that I did. I can't remember exactly when I did this. But these top three wells are a standard curve. So you can generate a standard curve, and using the standard curve, you can quantify exactly how much protein or how much antibody is present in the wells. Okay. Now, interestingly, when you add the substrate and you get that colorimetric reaction, it doesn't turn yellow. It actually turns a blue color. So what happens is you'll let the reaction proceed for a certain amount of time, and then you need to stop the reaction. We stop the reaction by adding a dilute solution of sulfuric acid. So sulfuric acid is very low pH. What happens to proteins when we expose them to low pH? Denature. Okay, so after a certain amount of time, we want to stop this reaction so it doesn't proceed any farther. So when we add the sulfuric acid, it's going to denature the enzyme, stop the reaction, and also it turns the color from blue to yellow when it does that as well. It's fine. We can still read the yellow color just fine on the ELISA plate reader. Okay? And depending on the intensity of the yellow color, that'll correspond, in an indirect ELISA, that'll correspond to how much antibody is present, directed against the antigen. Okay, so let's talk about another type of ELISA, a sandwich ELISA. Pardon me. So in a sandwich ELISA, we are detecting how much protein is there this time. Okay, remember, indirect ELISA, we're detecting if there's antibody against a certain antigen. In sandwich ELISA, we're detecting how much protein is there. Okay, so again, in the first step, we coat the wells. In indirect ELISA, we coated the well with antigen. In sandwich ELISA, we coat the wells with antibody. Okay, so for example, in this example, they're using a monoclonal antibody coating the wells. Second step is where we add our protein sample. If there's protein present in our protein sample, of course, the monoclonal antibodies are going to bind to it. Remember, because we're using monoclonal antibodies, these are very specific. They only recognize one particular epitope on the protein. Next comes the sandwich part of the sandwich ELISA. The sandwich part, again, is a secondary antibody. The secondary antibody also recognizes the protein, a different epitope on the protein so that it can get to it. So we're using, in this case, the secondary antibody would also be a monoclonal. Secondary antibody, again, is conjugated to an enzyme. So last step, we add substrate, and if the enzyme is present, then we're going to get a colorimetric reaction, and the intensity of that colorimetric reaction is going to be directly proportional to how much protein is bound in that well. Okay, so looking at the third bullet point, the bound monoclonal antibody binds antigen. The second antigen-specific antibody that is conjugated to an enzyme is added. The amount of antigen present can be determined by measuring the colorimetric reaction of the enzyme and substrate. Okay, so again, it's called the sandwich ELISA because our protein that we're trying to measure is actually sandwiched between the antibodies that we coated the well with and the second antibody that recognizes the antigen and is also conjugated to the enzyme. Okay, is everybody good with ELISA so far? Okay, good. So again, one important thing to remember is when we're doing indirect ELISA, we're trying to see if there's antibody present to the antigen that we coat the well with. So we're measuring antibody rather than protein. In a sandwich ELISA, okay, sandwich ELISAs are where we're actually measuring the amount of protein. That's an important point to keep in mind. Another way we can use antibodies to detect protein is something called a Western blot. So in the first stage of a Western blot, proteins are resolved on an SDS page gel and transferred to a nitrocellulose membrane. So we talked a little bit about SDS page before. So in SDS page gels, we separate out proteins based on size. So here's our polyacrylamide gel. Now, usually we wouldn't stain a polyacrylamide gel that we're going to use for a Western blot, but they're just showing it as stained so that we can see the protein bands. Okay, so the first thing we want to do, so we have our polyacrylamide gel, we've run out the proteins. So we have largest proteins on the top, smallest proteins on the bottom. In order to do a Western blot, we have to take the proteins on that gel and transfer them to a nitrocellulose membrane. Okay, so typically we'll take a nitrocellulose, cut it to the correct size, put it right against the polyacrylamide gel, and then there are some special machines that use electrophoresis to transfer the proteins from the gel to the nitrocellulose membrane. Okay, so that's where we are here. We have a polymer sheet. This polymer is usually made out of nitrocellulose. One thing that's important to know about nitrocellulose is nitrocellulose binds proteins irreversibly. Okay, so once they're transferred to the nitrocellulose sheet, they're there for good. In the next step, we take our nitrocellulose membrane that contains our proteins from our SDS page gel, and we incubate them with our antiserum. Okay. If the protein of interest is located in that polyacrylamide gel, the antibodies are going to bind to the band in that gel that contains those proteins. So in this example, these antibodies are recognizing this particular band that contains our protein of interest. Okay? Now again, we need to be able to visualize this, so we need a secondary antibody to visualize this. So again, we're going to take a secondary antibody. If we're probing this gel with rabbit antibodies, okay, we're going to need goat anti-rabbit. Okay, if we're using human antibodies, of course, we're going to need goat anti-human. But let's just say we're using a rabbit polyclonal serum in this case. So our secondary antibody would be like, for example, goat anti-rabbit HRP conjugated. That means they're going to stick to rabbit antibodies, and they're conjugated to horseradish peroxidase. Okay. Then we can take our nitrocellulose membrane, incubate it with a substrate, and if our antibodies are bound to a band on that nitrocellulose membrane, it's going to turn that band blue. Okay, we're going to have a colorimetric reaction, and we're going to turn that band a particular color. Usually in Western blots, it's going to turn it like blue or a dark purple color. Okay, so then we would be able to visualize our band. Okay. Just clear this. All right, I skipped over one question. So after we transfer the proteins to a nitrocellulose membrane, let me get my drawing tool back here. The nitrocellulose membrane needs to be blocked with a solution of dry milk proteins in an isotonic buffer. And I'm asking a question, why? Now, those of you, I know there's quite a few of you who took cell biology last semester, and we did some, well, we didn't do any SDS page gels, but we did some quantitative dot blots. Do you remember why we had to block our quantitative dot blots with dry milk?
No other proteins will bond to it.
Yeah, well, what are antibodies made out of? Protein, okay? And nitrocellulose binds protein irreversibly, right? So we need to block all the binding sites on the nitrocellulose that would bind the antibodies. Okay, and we can do that with just by using dry milk. Okay, dry milk is cheap. You can get it anywhere. You can get it at Kroger, and it works very well in this application. So you take an isotonic buffer like PBS or TBS, you just add like 1% or 2% dry milk, incubate your nitrocellulose membrane, and the milk proteins are going to bind to and occupy the protein binding sites on the nitrocellulose. Then we can go in and add our secondary antibody. If we, our primary antibody rather, if we added our primary antibody before this blocking step, then the antibodies are just going to bind everywhere non-specifically because they're made out of protein. Okay. Does this make sense to everybody? Okay, good. Okay, so after the blocked membrane is incubated with a primary antibody, for example, we can use a rabbit antibody. The membrane is washed and incubated with a secondary antibody. So again, the secondary antibody is the anti-antibody antibody, so to speak. So it's an antibody that sticks to our first antibody. Of course, that's conjugated to a membrane, and we can detect whether the band is there by a colorimetric reaction. Okay, this is an example of a Western blot that I did more years ago than I care to remember. I did this back when I was a postdoc in a lab I was at the University of Miami in Ohio. I was working in a microbiology lab. Interesting thing about this is we don't always have to use HRP as the enzyme that's conjugated to the secondary antibody. In this example, in this lab, we used an antibody that rather than giving a colorimetric reaction, it caused the chemiluminescent reaction. Okay, so it would actually cause the band to glow a little bit. And what we would do is we would take a piece of photographic film and put it against our nitrocellulose membrane, and then the film would be exposed. And that's actually what you're looking at. This is a piece of photographic film. I had this blot this morning. I still have this somewhere. I might have left the house without it. I'll try to remember to bring that so I can show you guys. But anyway, I won't go through exactly what the conditions of this, pardon me, the conditions of this experiment, what they were, but you can see that we got some nice bands at least. So you can see there's some pretty clear bands here and here. This antiserum, actually, I raised this antiserum that was used in this Western blot in a rabbit. So I made the recombinant protein, purified the protein, injected the rabbit, and then used it in a Western blot. So I did most of the work here, and you can see we got some nice bands. And again, I don't want to get too far in the weeds explaining exactly what the experiment was and what we're looking for. But yeah, I think that's a good example of a Western blot. All right, fluorescence microscopy. I don't think I have enough time to get into this. Well, it's 8:46. Yeah, I don't want to get into this till Friday. So let's stop here for today. On Friday, we'll continue talking about fluorescence microscopy, pardon me, immunofluorescent microscopy. But for now, we're done. So if there's no questions from Zoom, I'm going to stop the meeting and stop recording. And I'll see you guys Friday morning.